This video is going to be a review of our first standard chemistry of life for MCAS, and it's going to be reviewing enzymes specifically. This review should also help you with the first section of your MCAS review packet. First off, an enzyme is a protein. Remember from our review of macromolecules that one of the functions of a protein is to speed up reactions. Enzymes specifically are proteins that speed up chemical reactions. One thing that should help you on the MCAS is to remember that most enzymes end in the three letters ASE. The only exception to that we studied was pepsin. Let's take this chance to review a little bit of anatomy. Since we just studied anatomy, what are some enzymes in our digestive system? Hopefully you can remember that enzymes we talked about in digestive system were lipase, pepsin, amylase. And we didn't talk about it, but there is an enzyme in our digestive system called nuclease. Remember, lipase breaks down lipids, pepsin breaks down proteins, amylase carbohydrates, and nuclease actually breaks down nucleic acids. Now that we have a general understanding about what an enzyme is, let's talk about how they work. So let's say we're talking about amylase, which breaks down carbohydrates. Maltose is actually a carbohydrate. And if we say this is amylase, then what an enzyme does is it allows the carbohydrate maltose to attach on to the enzyme at something called the active site. It then binds to the active site. It should fit in perfectly. When we initially talked about enzymes, we talked about a lock and key model. So one important thing to remember is that enzymes are specific, which basically means they can only fit one substrate. So going back to these steps, if a different shaped molecule tried to fit in, let's say a protein, what would happen is the protein wouldn't be the right shape, so it would just bounce off. So for steps, we have a substrate, which is what the thing attaching is called, like carbohydrates, binds onto the active site, this area right here. Once it attaches on, a reaction takes place, and the products, in this case, since our enzyme was amylase, glucose, can leave. So the next thing to review about enzymes is what they do. Basically, enzymes lower activation energy. What that means is, if on our y-axis we have amount of energy needed, the graph that represents a reaction with an enzyme needs less energy than the red graph without an enzyme. You need to put less energy in when you have an enzyme. Last thing we're going to talk about is what types of things can affect enzymes. So enzymes work best at something called their optimal, meaning best, temperature, and optimal, pH. Usually, optimal temperature and optimal pH are going to be what the enzyme's typically found in. So for instance, pepsin is found in the stomach. The stomach is full of acid. So pepsin probably has a pH that's best at low pHs. If temperature gets too high, so if we look at the graph here, we can see that the rate of reaction increases in this graph until the optimal temperature is reached. Optimal temperature, highest point in the graph, is around maybe a 41, 42 degrees. Once that temperature is reached, though, the enzyme will denature, which means start to break down because it's too hot, and then all of a sudden the enzyme doesn't work as well. Try this problem just to see if you understand those key concepts. Figure out the optimal pH of amylase, and then use that information to predict the pH in the human mouth. The first thing you would need to do this problem is look at the key. you see that amylase is the solid line, because it says it right here. You don't need to concern yourself with this graph, because that's pepsin. To figure out optimal, you go to the highest point in the graph, you drop it down, you say optimal pH is around 7. Since amylase we know is found in the human mouth, that must also typically be around a pH of 7. At this point you've finished the key concepts for enzyme review. You can go ahead and do your review packet and rewatch parts of the video if you can't find the answers or don't remember parts of the video that are in your review packet.